Hello everyone, my name is Sanjeev Dahal. I'm a postdoctoral fellow in the Yang Lab at Queen's University. Thank you everyone for being here and listening to my talk. I'm grateful to ISCB for hosting this conference and providing me with an opportunity to present a new project I'm working on, which is using genome-scale model of metabolism and macromolecular expression to study biofilm development in Pseudomonas aeruginosa PA14. Cystic fibrosis or CF is a genetic disorder which leads to respiratory infection. According to Cystic Fibrosis Foundation Patient Registry, there are more than 30,000 people living with cystic fibrosis in the United States, and approximately 1,000 new cases are added every year. Because of thick and sticky mucosal lining, CF lungs become more susceptible to bacterial infections. One such bacteria is Pseudomonas. Pseudomonas is an opportunistic pathogen and one of the leading causes of hospital-acquired infections. In hospital-acquired infections within hospital setting, when patients come in contact through contaminated surfaces or reservoirs, Pseudomonas can be transmitted. Once Pseudomonas establishes itself, usually through biofilms, it is extremely difficult to eradicate from CF lungs. One reason why they are such a nuisance is because they are highly resistant to antibiotics. One mechanism of antibiotic resistance is biofilms. Biofilms are congregation of cells within a matrix composed of DNA, polysaccharide, and other compounds. They make bacteria more tolerant to antibiotics, help bacteria develop immune resistance, and provide a framework for collective behavior. Within biofilms, extracellular products such as siderophores, nucleotides can be used as public goods. That is, products that are secreted by one member of the biofilm can be consumed by another member of the biofilm. For our project, we are interested in the role of phenozines in biofilm development. Phenozines are naturally produced heterocyclic compounds which bring colors to microbial cultures. They are known virulence factors such as biocinin. Studies have also demonstrated their role in redox metabolism and in extracellular electron shuttle. Research has demonstrated that these molecules can be reduced within the cell. Then the reduced molecule can be transported out of the cell into the extracellular, where they get reoxidized by reacting with oxygen to produce reactive oxygen species, as can be seen here in the figure. Then the oxidized phenazine can be imported back into the cell for another redox cycle. The extracellular electron shuttle has been proposed to be involved in the survival of Pseudomonas during hypoxic conditions, such as those encountered within biofilms. In fact, phenazine mutants show different colony morphology compared to that of the wild type, as can be seen in the figure here. Phenazine mutants produce wrinkly colony phenotype, whereas wild type produce smoother colonies. We are interested in the role of biocinin in the development of two major components of biofilm of Pseudomonas aeruginosa. These components are PEL exopolysaccharide and extracellular DNA or eDNA. Biocinin is known to bind and activate RMCA, which is a phosphodiesterase and is involved in the degradation of C-diGMP. C-diGMP can allosterically bind and activate PEL-D enzyme, which is required for PEL polysaccharide biosynthesis. This suggests potential role of biocinin in inhibition of PEL synthesis. In fact, when the genes encoding the proteins involved in production of phenazines were deleted, the phenazine mutant was able to grow, and it overproduced pill exopolysaccharide. Biocyanin is also known to produce reactive oxygen species during redox metabolism. Reactive oxygen species are known to cause stress-induced cell lysis, which is a dominant route for eDNA release in Pseudomonas. This indicates potential role of biocyanin in the acceleration of eDNA release. In fact, multiple studies have shown that increase in biocyanin and stress or hydrogen peroxide leads to cell lysis through prophage induction. This brings us to our proposed hypothesis. The biofilm composition of Pseudomonas, which contains eDNA and PEL as major components, can be altered by changing the concentration of biocyanin. This elegant process provides Pseudomonas with mechanisms to survive in multiple harsh conditions. These mechanisms can be changed in biofilm properties such as overall charge or viscosity. This process improves the overall fitness of Pseudomonas. To study Pseudomonas biofilms, our lab utilizes numerous computational tools, including genome-scale models. Traditionally, these models could be used to predict metabolic state of a cell and were called M models. Within this framework, if one can visualize a cell as a network of interactions between metabolites, such network can be decomposed to a matrix. In this matrix, rows represent metabolites and columns represent reactions. This matrix can be studied at steady state using mathematical optimization methods such as constraint-based methods. Since the number of reactions are greater than number of metabolites, these systems are underdetermined systems, and this leads to multiple solutions satisfying this steady state assumption. Therefore, one needs to add constraints such as mass balance constraints and flux bounds to find the optimal solution at steady state. In reality, there are also processes occurring within the cell other than metabolism. Therefore, genome-scale models of metabolism and macromolecular expression, or ME models, were developed. These can model processes such as transcription and translation in addition to metabolism. Now, in addition to prediction of metabolic state, proteomic state of a cell can also be computed using ME models. Therefore, ME models are ideal tools to predict the proteomic allocation of resources towards metabolic activities. We began our work by first retrieving the genome-scale metabolic model from Papin Lab website. The model was created by Jennifer Bartel, and the study was published in 2017. Onto this model, we added biocyanin-related redox pathways. First, biocyanin and other phenazines like phenazine 1-carboxylic acid or PCA are known to be reduced abiotically in the presence of electron donors such as NADH. NADPH might also be an electron donor, but we are still gathering knowledge for this cofactor. Likewise, studies have shown that pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase can reduce these phenazines in the presence of their respective substrates. Furthermore, we have also added the reactions representing the extracellular electron shuttle as visualized in the figure. Two sources of reactive oxygen species are extracellular oxygen reduction by phenazines and intracellular NADH oxidation. By adding these reactions alone, we have coupled biocyanin with piocynin and other phenozines with central metabolic and redox pathways. Now let's see what the model can be used for in its current state. After adding these reactions, we have performed simulations to compare with the original model. We first wanted to compare whether the updated model can predict piocynin synthesis. Studies have demonstrated that piocynin synthesis or release rates increase exponentially during early stationary phase when the growth rate is slowing down. This can be visualized within the highlighted oval in the figure. When we performed the simulations using the original model, it could not predict piocynin synthesis at different growth rates, whereas the new model was able to qualitatively predict that as the growth rate increases, piocynin synthesis rate slows down. Since piocynin metabolism is associated with the generation of reactive oxygen species such as hydrogen peroxide, next we wanted to determine whether the updated model can predict growth-dependent hydrogen peroxide production as suggested by the experimental results in the study published in 2012. The model can qualitatively show that at higher growth rate, hydrogen peroxide levels are low. This supports the experimental results. At present, however, the model suggests that the primary source of hydrogen peroxide might not be piocynin. We have derived rate constants for reactive oxygen species generation from experimental data in the literature, but these parameters need to be scrutinized further. In the meanwhile, the model allows us to compare alternative sources of reactive oxygen species generation in different media conditions. This is an incredibly exciting new project that we intend to extend further. We plan to expand the metabolic model to the model of metabolism and expression, or ME model, so that we can couple the phenazine associated pathways with the production of biofilm matrix components such as PEL exopolysaccharide and eDNA. This expansion will allow us to test our hypothesis that cells, by tweaking the production of piocynin, can change the expression of pathways that lead to the alteration in the composition of biofilms. This control process, we believe, improves the overall fitness of Pseudomonas aeruginosa. We expect that with this study, we can find new insights into how we can treat Pseudomonas aeruginosa infections by effectively targeting its biofilm. Furthermore, we intend to couple genome-scale model with reactive transport framework and individual-based modeling. By doing so, we can study the cells within the temporal and spatial context of biofilms. For instance, within the spatial context, the cells closer to the air surface experience higher oxygen intake than those far away. Therefore, this framework provides us with insights into the heterogeneous metabolic states of the biofilm. Finally, I would like to acknowledge everyone in my lab. First, I would like to thank my advisor, Dr. Lawrence Yang, who gave me this opportunity to develop this project. He has been helping me throughout this process. I also want to acknowledge my considerate and helpful lab mates, Jiao and Hao. Jiao is helping me couple individual-based model with genome-scale model of Pseudomonas aeruginosa. Finally, I would like to thank everyone here who patiently listened to my presentation. I hope everyone is keeping safe and enjoying the conference. I would also like to apologize if I butchered anyone's name during the presentation. And with that, I am happy to take any question. Thank you.